Hello, this is me Shazad Kalado, and today we discuss about Brucella and brucellosis. First of all, history. The brucellosis was first discovered in 1850 in Malta. In 1886, David Bruce, a British army surgeon, isolated a cocobacillus that he named Micrococcus melitensis. Species. There are many species of Brucella, but the most important are explained below. Brucella melitensis, Brucella abortus. Now come towards Brucella abortus. Brucella abortus is gram-negative bacterium. It is contagious, costly disease of ruminant, for example, cattle, bison, and cervets. Here you can see that in picture. Brucella melitensis. Brucella melitensis is a gram-negative coccobacillus bacterium, mostly commonly acquired from goats and has been acquired from sheep and camels. Morphology. Shape. Small cocobacilli, gram-negative, non-motile, aerobic and non-capsulated. Here you can see that in picture. Color. Pinkish reddish color. Size. Its size is 0.6 to 1.5 micrometer. Cultural characteristics. Brucella are strict aerobes. Brucella abortus is capnophilic, many strains requiring 5 to 10% of carbon dioxide for growth. Optimum temperature is 37 and pH is 6.6 to 7. Grow on simple media, though growth is slow and scanty. Isolation of Brucella requires a selective media. This contains bacitracin, nalidixic acid, cyclohexamide, nystatin, polymyxin B, and vancomycin in a serum dextrose agar base positive in about 50 to 70% of cases. Bone marrow culture standard. Samples of any body fluid or tissue can be cultured. Blood most common, incubated at 37 degrees centigrade in air, enriched with 5 to 10% of carbon dioxide. Typical virulent Brucella colonies are smooth and translucent. Merit gold standard. Demerit hazardous bacterium. Level 3 laboratories. Labor intensive, time consuming procedure. Mode of infection. The most common way to be infected is by eating or drinking with the bacteria. Use of infected syringes and contact with infected birthing tissues and fluids, for example, placenta, aborted fetuses, fetal fluids, vaginal discharges. Here you can see that in picture. Pathogenesis. Here is the whole pathogenic cycle data how these pathogens infect the host. Brucellosis clinical presentation. Abortion between the 5th and the 7th month of pregnancy, stillbirth, birth of weak calves, retained placentas, reduced milk production. Calves from infected cows may have latent infections, decreased fertility, poor conception rates, and occasionally enlarged arthritic joints. Biochemical tests. Catalase test, milk ring test, and number third is Rose Bengal plate test. Diagnosis. Serology. Another is hypersensitivity test, and number third one is ELISA. Milk ring test. Frequently used test, a drop of colored Brucella antigen, Brucella abortus or Brucella melitensis with hemotoxylin is added to milk in a test tube, incubated in a water bath at 70 degrees centigrade for 40 to 50 minutes. Positive, blue ring at the top leaving the milk unstained. Negative, no ring, milk remain uniformly blue. Rose Bengal plate test. Test antigen, heat inactivated Brucella abatus cells stained with rose bengal and buffered at pH 3.65. Place 30 microliter of test antigen next to 30 microliter of serum on a flat, white or transparent surface. Mix the drop with a stick, producing a zone of 2 cm diameter. Rock the slide gently for about 4 minutes. Formation of agglutinates. Positive confirmation of result by complement fixation test. And if the result is no formation of agglutination, then it is negative.